the fascinating world of the genes with Rohini, and today we are going to learn about the stringent response, the response given by the bacterial cells. This stringent response, it is given when there is the arrival of the starvation conditions or poor growth conditions. Particularly when there is a depletion of the amino acids in the medium, at that time there is reduction in the protein synthesis, carbohydrate synthesis, lipid synthesis and the nucleotide synthesis, and majorly and particularly at a higher level there is a reduction into the synthesis of the rRNA and the tRNA, and this it is called as a stringent response. So, how a cell recognizes that there is the arrival of the starvation conditions, there is a limitation of the amino acids, and now a cell it has to give the stringent response. Before that, we have to memorize that what is going to happen into the elongation during the protein synthesis. Here it is the ribosome, it is the mRNA, and the ribosome there is a P site at the A site. Then there is the presence of the peptidyl tRNA at the P site. At the A site there will be the placement of the charged tRNA. Okay, the next step it is what? In the next step there is the transfer of this peptide to this amino acid. That is the peptide from the peptidyl tRNA, it is going to be transferred to the amino acid at the charged tRNA at the A site. And after that peptide bond synthesis, after that there is a translocation, and again the process it is going to repeat. Now, when the conditions they are the starve conditions, okay, when the conditions they are the starve conditions, at that time there is lower levels of the charged tRNA and higher levels of the uncharged tRNA, and during these conditions the uncharged tRNA it can come and it can bind at the A site. Okay, the uncharged tRNA it is going to come and it is going to bind at where? At the A site. It is going to be placed at the A site. So as it is uncharged, okay, as this tRNA it is uncharged, there will be no peptide bond formation. Peptide transferase it is not going to work because there is no amino acid at this tRNA at this A site. So what is going to happen? No peptide bond formation, no translocation it will be there. Means what? This process, this protein synthesis process, it is going to be halted due to the presence of the uncharged tRNA at the A site. Okay, now this it is going to be recognized. Okay, this it is going to be recognized. What? The uncharged tRNA it is at the A site. It is going to be recognized by the conformational changes into the L11 protein. Okay, which protein? L11 protein. The L11 protein which is present into the ribosome. Okay, it is a ribosomal protein. So there is the conformational changes into the L11 due to what? Due to this uncharged tRNA placed at the A site. Okay, and this L11. Okay, now this L11. Now it is going to activate. Okay, it is going to activate the protein which is present into the ribosome only, and that protein it is a RelA. RelA it is also called as the stringent factor. What it is called? It is also called as the stringent factor. So the RelA, okay, the RelA it is getting activated, and when this RelA it is getting activated, at that time, at the same time, this uncharged tRNA from the A site it is going to be removed. Now, what is the function of this RelA? The another name for the RelA, it is guanine pentaphosphate synthetase. What it is? It is guanine pentaphosphate synthetase. Means the RelA, what it is going to synthesize? Guanine pentaphosphate. What it is going to synthesize? Guanine pentaphosphate. So here ATP plus GTP, it is going to give the guanine pentaphosphate, and with the help of the ribosomal protein, slowly it is going to convert it into the guanine tetraphosphate. Okay, so these are the two signal molecules. These are the two molecules which are synthesized, and these molecules now they are called as the alarmos. Okay. This guanine tetraphosphate, it can also be synthesized from the ATP plus GDP in the presence of the another proteins which are present into the ribosomes. Okay, so this it is the minor pathway, and this it is the major pathway. Okay, so ATP plus GTP in the presence of RelA stringent factor, it is going to give you the guanine pentaphosphate. Okay, and ATP plus GTP in the presence of some of the ribosomal proteins, it is going to give you the guanine tetraphosphate, or the guanine pentaphosphate. Slowly it is going to convert into the guanine tetraphosphate. Now these are the negative regulators. Okay, these are the two negative regulators, and these two negative regulators, what they are going to do? As they are negative regulators, what they are going to do? It is going to negatively regulate any operon. Okay, that it is going to switch off the operon, and which is that operon which is going to be switched off by these alarmos? That operon, it is the rRNA operon. Which operon? The rRNA operon. And as the rRNA operon, okay, it is going to be switched off, the concentration of the ribosomes into the cell, now it is going to be lower down, and ultimately the rate of the protein synthesis, it is also going to be lower down, and the another metabolic activities, they are also going to be lower down. Okay, so in this way, there is the response given by the bacterial cell in the absence or in the lower levels of the amino acids, and that response it is called as the stringent response. Now, whenever there is the incoming of, again return of the favorable conditions, at that time what is going to happen? The protein synthesis rate, it is going to be increased. That is, all the synthetic processes, all the synthesis, it is going to be at a higher level. But how? What is going to happen to these negative regulators, guanine pentaphosphate and guanine tetraphosphate? As there is the incoming of the favorable conditions, at that time these alarmos, it is going to be cleaved by the protein, and that it is the SPOT. Okay, who is going to cleave this? The SPOT, it is going to cleave these alarmos, and again there will be the high rate of the synthetic processes into the cell. Okay, this it is regarding the stringent response. If you have any doubts, if you have any comments, you can write down into the comment box. If you like my video, please subscribe my channel and also share my videos with your friends so that the knowledge it is going to spread. Thank you.